KPG Arts, Commerce and Science College, Igatpuri, district Nashik. And in today's lecture, we are going to discuss about the cell division in living systems. So there are two different kinds of cell division in all the living systems, which are mitosis and meiosis. So mitosis takes place in the vegetative cells. Vegetative cells means which are not directly involved in sexual reproduction. And second type of cell division is the meiosis. Meiosis takes place in the germ cells, which directly play a role in the sexual reproduction in living systems. That may be plants, animals, or any kind of microorganisms. So this is the cell cycle of eukaryotic cells. We know that eukaryotic cells are modern cells having different kinds of compartmentalization, like the nucleus, chloroplast, mitochondria. So such kind of cell organelles are there inside the eukaryotic cell. And these eukaryotic cells follow which kind of pattern, so that we are going to learn in this PPT. So cells, they grow, cells they may enlarge and they may be divided by means of vegetative method, that is the by means of mitosis or meiosis. So inside the cell there is a genetic material in the eukaryotic system which is present inside the nucleus, which is known as DNA. So this DNA got replicated. Replication means DNA makes the copy of itself. So mitotic cell division, it produces the daughter cells which are identical to the parental cell or mother cell. So why they are identical? Because they follow the mitotic cell division. The timing of replication and the cell division is highly regulated in this kind of cell cycle. So there are three different kinds of phases of cell cycle. One is G1 phase, then S phase, and second one is G2 phase. So G1 and S phase together they include in the interphase of cell cycle. So in the G1 phase, whatever the genetic material which are going to be replicated at the S phase, that is synthesis phase, DNA got synthesized here. So what are the requirements of DNA replication? For example, enzymes are there, different kinds of molecules are there, different kinds of the elements like magnesium ions are there to catalyze that reaction. Those requirements they got fulfilled in the G1 phase. If all the requirements will be fulfilled, then and then only cell will go into the next phase, which is known as S phase. So S phase stands for synthesized phase, which synthesizes the DNA. Okay. So here the DNA replication will take place, and if the DNA replication is completed, then cell cycle will going to move towards the next phase, which is known as a G2 phase. So in the G2 phase, the DNA replication errors they got corrected. Okay, so if there is a kind of errors in the DNA replication, so that will be corrected during the G2 phase, and if all the errors, if they will be get corrected, then and then only cell will going to move towards the next phase, which is known as a M phase. So M phase is very important because this is the last step of cell cycle. M stands for mitotic. So here mitosis or meiosis will take place. So it depends on which kind of cell it is. So if this cell is a vegetative cell, which is not directly involved in sexual reproduction, then this cell will going to follow the process of mitosis. And if it is a germ cell, which is a reproductive cell, then cell will going to follow the meiosis process. The main stage of the cell cycle is the gap 1 phase, synthesis phase, gap 2 phase, and mitosis, which is discussed earlier. So this is the gap phase. Next one is the synthesis phase. This one is the G2 phase, and this is the mitosis, which we are going to be focused in today's lecture. So in the interface, we have seen here the structure of plant cell. Okay, so I am generally belongs to the subject botany, so that's why I will give the example of plant cells here. So this is the cell wall, the nucleus, the genetic material present inside that. And this is the actual cell which are observed under the microscope. So mitosis is the process where this process is divided into the different kinds of stages. First phase is the prophase, next phase, that is second phase, is the metaphase, third phase is the anaphase, fourth phase is the telophase, and which is all these four phases followed by the cytokinesis. So we will see each and every step in detail. First phase, that is prophase. So here you can see this is the cell. Inside that cell you will find the nucleus. Inside the nucleus you will find the genetic material. This is the nucleolus, okay, and these are the centrioles. So during the prophase, the genetic materials got condensed. The spindle fibers, they will going to be appear. Chromosomes are captured by the spindle fibers, what we can see here. So this takes place in the prophase. While in the metaphase, all the chromosomes, what you can see here, which are irregular, dispersed inside the nucleus, so they will going to be arranged at polar region, arranged at the central region, and from the polar region, south as well as north pole, the spindle fiber they will attach to the centromere of the chromosome. Next phase is the anaphase. So this spindle fiber, they will separate the sister chromatids of the chromosome, so that you can see here the chromosomes are moving towards the north pole as well as towards the south pole. So in this way the chromosomes, they were divided into these sister chromatids and move towards the polar region. This is the actual photographs of anaphase, what you can see here. Next one is the telophase. So in the telophase, the chromosomes are already moved towards the polar region at the north as well as at the south pole. Then the nuclear membrane will reappear, okay, and there is a separation of genetic material, separation of the chromosomes in the telophase. But still there is a common what we can say the cytoplasm which is not separated till this step. The final is the cytokinesis. So in the cytokinesis, cell plate will going to be formed in case of the plant cell, and in this way one cell will completely separated from each other by means of the formation of cell plate. After that there is a formation of cell wall. Thank you.